Hello everyone, Mr. Linder here. Let's continue our conversation about aerobic metabolism while looking at the citric acid cycle and the electron transport system. So aerobic metabolism is catabolic metabolism in the presence of oxygen. So in a previous video, we looked at glycolysis and the transition reaction. In this video, we're going to continue talking about the metabolic pathways in the citric acid cycle and then the electron transport system. The transition reaction yielded us two acetyl-CoA molecules. Those acetyl-CoA molecules came from glucose. So glucose went through glycolysis to produce pyruvic acid, and then pyruvic acid went through the transition reaction to produce those two acetyl-CoA molecules. When coenzyme A is removed, you're left with an acyl unit or an acetate molecule. That acetate molecule enters into what we call the citric acid cycle. And this is taking place in the mitochondrial matrix. So we're currently inside the mitochondria in the mitochondrial matrix. And so what happens is the acetate molecule, this 2-carbon subunit, reacts with oxaloacetate, also known as oxaloacetic acid. And when the acetate molecule and the oxaloacetic acid combine together, they form citric acid or citrate. So citric acid is the first molecule that's produced in the citric acid cycle, which is why it's referred to as the citric acid cycle. Oftentimes, too, it's also called the Krebs cycle. So 2-carbon acetate combines with 4-carbon oxaloacetic acid or oxaloacetate to form citric acid, also known as citrate. Notice a 6-carbon molecule. Once you produce citric acid, the first molecule in the cycle, then it's just a series of reactions that convert the citric acid back into oxaloacetic acid. So it's a cycle. We're just going to turn this all the way around. So from citric acid all the way back to oxaloacetic acid. Since there's 2 acetyl-CoAs produced in the transition reaction, then this series of reactions happens twice. So this is going to happen 2 times as we spin the citric acid cycle. Through a series of 7 reactions, we're going to see citric acid converted all the way back to oxaloacetic acid. The first reaction is citrate becoming isocitrate. The next reaction is isocitrate to alpha-ketogluteric acid or alpha-ketoglutarate. Notice that in that particular reaction, you are producing a high molecule NADH. So NAD has received electrons and, of course, hydrogens. But it's receiving those electrons, and so it's forming this high-energy NADH molecule. You'll also notice that a carbon dioxide is removed. From isocitrate to alpha-ketoglutarate, when you remove a carbon dioxide, you end up now with a 5-carbon molecule. Alpha-ketogluteric acid, or alpha-ketoglutarate, goes through a reaction in which you produce another high-energy NADH molecule and produce succinyl-CoA. That means that coenzyme A has been added. There's also been a removal of carbon dioxide, so now we're down to a 4-carbon molecule. Succinyl-CoA goes through a reaction to form succinic acid or succinate. The coenzyme A is removed. In that process, we're producing 1 ATP molecule. From succinic acid, we have the production of fumaric acid or fumarate. In that process, we produce a high energy molecule known as FADH2. From fumaric acid to malic acid, we have the conversion, again, from just one 4-carbon to the next 4-carbon, but it's been rearranged, so fumaric acid to malic acid. Then from malic acid to oxaloacetic acid, we get the production of another high-energy NADH molecule. So again, it's just a series of reactions, and it happens twice. So we're going to go through that series of 7 reactions 2 times. What do you get from the citric acid cycle? As you go through all of these reactions, what are the outcomes that you see? So the outcomes that you see are the production of 3 NADHs. Every time it turns, you get 3 NADHs. You get 1 FADH. And then you also produce 1 ATP. And of course, there's a removal of 2 carbon dioxides. Okay, so we lose 2 carbon dioxides. So since this spins twice, because you have 2 acetyl-CoAs entering into the cycle here, since it spins twice, then you actually have 6 NADHs, you have 2 FADHs, and you produce 2 ATPs. So again, not a lot of ATP production. We only got 2 ATPs from glycolysis. You get 2 ATPs from the citric acid cycle, but we're starting to add up all of these NADHs and FADHs from glycolysis, the transition reaction, and the Krebs cycle. And it's these high energy molecules that are now going to move into the electron transport chain. And then, of course, remember that the carbon dioxides have come off. So since you lose 2 for every turn of the cycle, you actually lose a total of 4 carbon dioxides. So there were 2 from the transition reaction and 4 from the citric acid cycle. So that's our 6 carbon dioxides that are produced as waste in aerobic metabolism. Now that we have all of these NADHs and FADHs, we can move into the electron transport system, or the electron transport chain. This is the final process in actually producing the maximum amount of ATP from aerobic metabolism. So the electron transport system is going to take these NADH and FADH molecules. They're going to donate their electrons, and we're going to use that energy from those electrons to power the electron transport system and produce ATP. So where is the electron transport system taking place? Well, here we see the Krebs cycle, or citric acid cycle, was in the mitochondrial matrix. The electron transport system is a series of proteins that are found in the inner membrane of the mitochondria. So we're in the, what we call the cristae, and we're in the cristae of the mitochondria. So this is that inner mitochondrial matrix. And this is, inner mitochondrial membrane next to the mitochondrial matrix. And so these are proteins embedded in that membrane that are going to be involved in the electron transport process. The electron transport proteins, this will give you a better idea of what they look like. The electron transport proteins embedded in that membrane are proteins like the FMN protein. We have the coenzyme Q protein, and then we have a series of cytochrome proteins. So here we see cytochrome B, here's cytochrome C, cytochrome A, so you get the idea. So FMN proteins, coenzyme Q proteins, and the cytochrome proteins. Coenzyme Q is pretty famous these days. You may have heard of Qunol supplements. Qunol supplements have coenzyme Q, coenzyme Q10 in there, and they're used in this electron transport system so that we can produce ATP. Each of the proteins in this inner mitochondrial membrane are involved in transferring electrons. Now you'll notice that the NADH molecule transfers its electrons to the first protein complex, so to this FMN complex. FADH2s transfer later in the process. So the energy yield from NADHs is different than the energy yield from FADHs. You actually get more ATP from an NADH than you do from an FADH. And that's just because of where you're transferring the electrons. So there's less hydrogen ion transport. So we're going to look at how this actually works. So electrons are transferred to these proteins within the inner mitochondrial membrane, and those electrons are going to move along these proteins, powering those proteins so they can pump hydrogen ions. So let's take a different view of what's going on here, and then we'll come back to what it looks like in the membrane. But here we have this idea that NADH, right, and FADHs, so FADHs, NADHs and FADHs can transfer electrons, okay, to the mitochondrial proteins, and that's going to help us in the process of producing ATP. So here we have the FMN protein, the coenzyme Q, the cytochrome proteins. And so depending upon where you transfer those electrons will determine how much energy you can get. So since NADHs transfer higher up in the process, you can produce more ATP along the way. Since FADH transfers in lower in the process, you're going to produce less ATP along the way. What you'll also notice is that as the electrons are being transferred through this process, they eventually need to end up somewhere. So those electrons are going to combine with oxygen, right, and bring hydrogen along. And so you produce water as a product of the electron transport system. So oxygen, right, this is aerobic metabolism, and so this is finally where oxygen comes into play. Aerobic metabolism uses oxygen. Oxygen acts as the final electron acceptor. So oxygen is the final electron acceptor. Without oxygen, these electrons would back up the electron transport system, and then we wouldn't be able to transfer electrons from NADHs and FADHs anymore. So you have to have oxygen as the final electron acceptor. So let's look at this as a full process. Okay, so if we were to zoom in to what the electron transport system is doing, or what the electron transport chain is doing, we can follow along in this picture. So what's happening is these high energy electrons are coming from NADH or FADH. And remember that depending upon which molecule it is, it transfers those electrons to different locations. So the NADH electrons are being transferred earlier in the process. The FADH electrons are being transferred later in the process. So NADHs give us more ATP than FADHs do. FADHs give us less ATP. And we'll look at that in the final totals when we add up how much energy we actually get. So these high energy, though, electrons are being donated to the electron transport system. So this is that FMN molecule. This is coenzyme Q, and then these are the cytochromes. I'm just going to put cyto just to label them. So these are the cytochrome molecules. So electrons are bouncing from protein to protein to protein in the electron transport system. So we dump the electrons. The electron pairs, now you can see in this diagram, those electrons now are moving, right, to these proteins, and those electrons are going to be passed from one protein right, to another. As you pass those electrons from one protein to another, you're going to see the movement of hydrogen ions. So hydrogens are being pumped, okay, from the mitochondrial matrix into this intermembrane space. And so this is the intermembrane space between the inner mitochondrial membrane and the outer mitochondrial membrane. So you're creating a higher hydrogen ion concentration in this space. The electrons continue to bounce along the system. The electrons get to the end of the system and they are added to oxygen. And so oxygen is the final electron acceptor. And when you add the electrons to oxygen, you will combine hydrogen ions with it. And this is where you get the production of water in the process. So water is being formed as you combine electrons and hydrogen ions with oxygen. Once you have a hydrogen ion concentration, so we've built up these hydrogen ions within that intermembrane space, what we have here is essentially what we call chemiosmotic theory. And what we're getting is, it's also referred to as oxidative phosphorylation. This chemiosmotic theory says that the potential energy that is created by the hydrogen ion gradient is going to be used to produce ATP. And the way it works is the hydrogen ions move through this enzyme. This enzyme is known as an ATP synthase. It synthesizes ATP. And so as the hydrogen ions move through the ATP synthase, you're going to produce ATP. For every 3 hydrogen ions, so for every 3 hydrogen ions that move through the ATP synthase, we're going to produce 1 ATP molecule. So electrons power the electron transport system. We move hydrogen ions into the intermembrane space. And then as the hydrogen ions move through the ATP synthase, we produce ATP energy. And that's how the electron transport system works. How much energy do we actually get then from all the different processes of aerobic metabolism? So what we want to do is we want to look at the overall yield for aerobic metabolism. So from glycolysis, from the transition reaction, from the citric acid cycle, how much energy can we actually produce? Okay? So what do we know from the processes? Well, we know that we got 2 NADHs from glycolysis. We know that we got 2 NADHs from the transition reaction. We know that we got 6 NADHs from the citric acid cycle. We also got 2 FADHs from the citric acid cycle. So what we can do, what we can do is we can calculate how much energy we're going to get. We also know that we got 2 ATPs from glycolysis, and we also got 2 ATPs from the citric acid cycle. So we've made 4 ATPs, but how much do we get from the NADHs and the FADHs? The NADHs give us 2.5 ATPs. Now, different sources, different resources out there will give you different numbers. But from research, we typically know that an NADH will actually only yield us 2.5 ATPs. Some resources will tell you that an NADH will give you 3 ATPs, and that's why you'll get different calculations for total ATPs. But we're going to go with NADH gives us 2 ATPs per molecule. FADHs give us 1.5 ATPs per molecule. they give us less energy because they dump their electrons at a different location in the electron transport system. So what do we have so far? Well, we know that we have 4 ATPs that were directly produced, 2 from glycolysis, 2 from the citric acid cycle. We have how many total NADHs? Well, we have 2 plus 2 is 4, plus 6, so that's 10. So if we have 10 NADHs, 10 times 2.5 ATPs would give us a total of 25 ATPs. If we have 2 FADHs that was produced from the citric acid cycle, and that's times 1.5 ATPs, then we know that that gives us another 3 ATPs. So if we go with those numbers, we have 25 plus 3 plus the 4 that we already made. So that would give us a grand total of 32 ATPs. So that's where this number comes from right here in this formula, okay? We get 32 ATPs if we just look at it this way. 4 ATPs that were produced, glycolysis and citric acid cycle. We got 3 ATPs from the FADHs produced in the citric acid cycle. And we get 25 ATPs from the 10 total NADHs. However, there are situations where if you look at cytoplasmic NADHs, so the cytoplasmic NADHs would be those produced from glycolysis. It typically takes energy to move those NADHs from the cytosol into the mitochondria. So a lot of estimates will say those NADHs are not going to yield you the full 2.5 ATPs per NADH. In essence, they're calculated based off of really the FADH yield. So what we're saying is that cytoplasmic NADHs may only yield you 1.5 ATPs per NADH instead of the total 2.5s that you normally get. So if that's true, you need to change your calculation just a little bit. So what we can say is the cytoplasmic NADHs, which there are 2 of them, okay, are only going to yield you 1.5, right, ATPs. So that would be a total of 3 ATPs from the cytoplasmic NADHs. That means you have 8 NADHs that are not cytoplasmic, that were made in the mitochondria. So that's times the 2.5. So that's going to give you 20 ATPs. You still have your FADHs, so you have 2 FADHs. That's times 1.5. So that's still 3 ATPs. And then you still have the 4 ATPs that you previously made. So if you add up those numbers, you've got 20 plus 6, that's 26, plus the 4, that's going to give you 30 ATP total. And so that's why it gives you a range of 30 ATPs to 32 ATPs. So what we're saying is one glucose, right, has the ability to give us anywhere from 30 to 32 ATPs, depending upon if you are reducing the amount of energy from the cytoplasmic NADHs or if you're just leaving them as a regular NADH. So there are different ways to calculate this. In essence, one glucose gives you 30 plus ATP, which is a really good yield when we're talking about aerobic metabolism in the presence of oxygen. If you're not doing aerobic metabolism, then you're going to have a much lower yield of energy. And then if you're looking at other resources, you may see numbers all the way up to, say, 34 to, say, 36 ATP. That just means they're using a different calculation for how much yield you get from NADHs and FADHs. So that is the citric acid cycle and the electron transport system and how much energy we get from those in aerobic metabolism. I hope that helps. Take care.